dear members of the Dissertation Council and the attendees, in accordance with the minutes of the Academic Council of the Kazakh National Agrarian Research University, number 5, dated December 2024, for persons we approved as permanent members of the Dissertation Council. In addition, by the minutes of the Dissertation Council number 4, dated March 20, 2026, temporary members and official reviewers were appointed. Permanent members of the Dissertation Council. First, Chairman of the Dissertation Council, Сағымбай Kaldıbayev, Doctor of Agricultural Sciences, Professor of the Department of Soil Science, Agrochemistry and Ecology, non-profit joint stock company, Kazakh National Agrarian Research University. Second, Deputy Chairman of the Dissertation Council, Bahidjan Dusenbekov, candidate of Biological Science, Professor, Chairman of the Board, Kazakh Research Institute of Plant Protection and Quarantine, named Jazken Jimbayev. Third, Scientific Secretary of the Dissertation Council, Gulsharia Nursapaevna Kayirova, candidate of Agricultural Sciences, Professor of the Department of Fruit and Vegetable Growing, Plant Protection and Quarantine, Kazakh National Agrarian Research University. 4. Member of Dissertation Council, Elmira Salnikov, candidate of Agricultural Sciences, Professor at the University of Belgrade, Institute for Multidisciplinary Research, Department of Plants, Soil and Nanosystems, Belgrade, Serbia. Temporary members of the Dissertation Council, Kanat Yermekbaev, Doctor of Philosophy, Head of the Laboratory of Molecular Genetics and Genomics, Jetesu University, named after Jansugurov. Vadim Hasanov, candidate of Biological Science, Professor of the Crop Production Department at the Institute of Agriculture and Forestry of the Sekensifulin Kazakh Agri-Technical Research University. Kazbek Dusenbayev, PhD, researcher at the Laboratory of Biodiversity and Genetic Resources, National Center for Biotechnology. Official reviewers, Елена Rahimova, Chief Researcher of the Laboratory of Mycology and Algology, Doctor of Biological Science, Associated Professor, Institute of Botany and Phyto Introduction. Allah Bakhsh, PhD, Associated Professor, Center of Excellence in Molecular Biology, University of the Punjab, Lahore, Pakistan. Okay, in accordance with the rules for awarded degrees of the Committee for Quality Assurance in Science and Higher Education of the Ministry of Science and Higher Education of the Republic of Kazakhstan, the quorum for holding the meeting is present. Permission is hereby granted to open the meeting of the Dissertation Council. All permanent and temporary members of the Council and official reviewers are in attendance. There is a proposal to commence the work of the Dissertation Council. Please vote. Who is for or against? Voted unanimously. The proposal is adopted unanimously. Today's agenda defense of the dissertation of Elmira Sovetovna Исмагулова on the topic phytopathological assessment of introduced walnut varieties and the development of protective measures against major diseases for the southern fruit growing zone of Kazakhstan, submitted for the degree of Doctor of Philosophy in the educational program AD08-104, Plant Protection and Quarantine. The dissertation was recommended for public defense at the extended meeting of the Department of Fruit and Vegetable Growing, Plant Protection and Quarantine of Kazakh National Agrarian University. Scientific consultants. Gulsharia Kairova, candidate of Agricultural Sciences, Associate Professor of the Department of Fruit and Vegetable Growing, Plant Protection and Quarantine, Kazakh National Agrarian University. Second consultant, Hussein Basim, Doctor, PhD, Professor, Head of Department of Plant Protection, Faculty of Agriculture, Akdeniz University, Turkey. The research on the dissertation topic was conducted at Kazakh National Agrarian University. Dear members and participants of the dissertation council, given that Gulsharia Kairova serves as the scientific consultant of the dissertation work of Elmira Исмагулова and accordance with clause 6.2 of the regulation on the dissertation council of the Kazakh National Agrarian Research University, the chairman, deputy chairman, and the scientific secretary of the dissertation council may not perform their duties during a meeting at which the dissertation of a doctoral candidate for whom they serve as a scientific consult is being considered, or where a conflict of interest exists. In such cases, the duties shall be assigned to the two other members by decision of the dissertation council. In this regard, I request that the duties of academic secretary be assigned to temporary member of the dissertation council, Kanat Yermekbaev, for the duration of the council meeting. If there are no objections, I ask you to vote in favor of this proposal. Against, none, abstained, adopted unanimously. The floor is given to acting scientific secretary of the dissertation council to announce the submitted documents. Please, Kanat.
Thank you very much. Good morning, dear members and the participants of the dissertation council. So doctoral candidate Elmira Sovetovna Исмагулова was born on November 18, 1993 in the city of Shymkent, Turkestan region. Academic background. Elmira Исмагулова completed her bachelor's degree between 2011 and 15 at Al-Farabi Kazakh National University with a specialization in hydrology. Code 5B060100. She subsequently obtained her MSc or master's degree between 2015 and 2017 from Kazakh National Agrarian University in ecology. Code 6M6060800. Then she pursued her doctoral studies between 2021 and 24 at the Kazakh National Agrarian Research University, hereafter mentioned as KazNaru, under the educational program in plant protection and quarantine. Professional experience from 2018 to 2022, she served as the plant protection agronomist at the Ecoculture Greenhouse Complex located in the village of Karatöbe, Karasay District, Almaty region. Submitted documentation in accordance with the requirements of the Committee for Quality Assurance in Education and the Science of the Ministry of Science and Higher Education of the Republic of Kazakhstan. Doctoral candidate Elmira Savetovna Ismagulova has submitted the following documents. First, application addressed to the Chairman of the Board, Rector of KazNaru. Reviews from domestic and international academic supervisors certified by their respective institutions. Third, positive conclusion of the extended departmental committee meeting. Fourth, dissertation in hardcover binding. Fifth, list of scientific publications. A total of 8 scientific publications works have been published on the dissertation topic, including 4 articles in journals recommended by the Committee for Quality Assurance in Science and Higher Education of the Ministry of Science and Higher Education of the Republic of Kazakhstan. 2 articles in Scopus Index journals at the 65th and the 88th percentiles respectively, and the 2 articles in international conference proceedings. 6, dissertation abstract in Kazakh, Russian, and English languages. 7, extract from the Academic Council minutes confirming the approval of the dissertation topic and the assignment of the academic supervisors. 8, copy of the transcript confirming completion of the doctoral professional curriculum. 9, certificate from the Scientific Library of KazNaru and the National Center for State Scientific and Technical Expertise, NCST, confirming publications in international scientific journals indexed in Web of Science or Scopus. 9, plagiarism check certificate for the PhD dissertation. 11, conclusion of the Ethics Commission of KazNaru. 12, copy of the order granting admission to the dissertation defense. The dissertation is presented in English language. All submitted documents and the materials comply with the degree awarding rules of the Committee for Quality Assurance in Science and Higher Education of the Ministry of Science and Higher Education of the Republic of Kazakhstan. Thank you.
Thank you very much. Are there any questions for the academic secretary? No? Okay. The floor is given to the application to present the substance and the main provisions of the dissertation. The allotted time is 20 minutes. Please adhere to schedule.
Good day, dear Chair, Dissertation Council, esteemed members of the Dissertation Council, and distinguished guests. It is great honor for me to present my scientific research today. The title of my scientific research is Phytopathological Assessment of Introduced Walnut Varieties and Development of Protective Measures Against Major Diseases for the Southern Fruit Growing Zone of Kazakhstan. So, allow me to start from the relevance of my work. Walnut has a high nutritional, medicinal, and economic value. Kazakhstan has favorable conditions for plantations growing, but we have several limiting factors. First limiting factors, it is a fungal and bacterial diseases. Second one, it is lack of registered pesticides in our country. So therefore, our farmers have a losses of yield, and we decide to take this research and investigate the field and laboratory experiments. At this slide, I want to tell about research objectives. The first, to conduct field monitoring and to determine disease incidence and disease severity index. Also describe the symptoms of pathogens in the field. Second one, to identify fungal and bacterial pathogens using molecular genetic methods. Third, to study the genetic diversity of walnut trees in Kazakhstan, cultivated, local, and wild forms using CNP analysis. The next one, it is evaluate the resistance of walnut varieties to the main identified pathogens. To evaluate the effectiveness of modern fungicides against major walnut diseases. And the last one, to develop recommendations for protecting walnut plantations from fungal and bacterial diseases. From 2022 to 2024, we conducted a field monitoring and surveys 9 farms. As you can see on the pictures, 3 farms from Turkestan region, 1 from Jambul region, and 5 from Almaty region. Our study included walnut varieties, wild forms, 23 introduced forms from France, China, Moldova, Romania, and Ukraine. For example, Chinese cultivar Liao are grown in Silkaley from Turkestan region. Local forms are grown in Saragash Jersiya from Turkestan region. Wild growing walnut grows in Alma Arasan Grove. In the Manshuk rural farms, 23 introduced cultivars from France, China, Moldova, Romania, Ukraine. At this slide, I want to tell how climatic condition can cause on the pathogen development. In the Talgar, Issyk, and Turgen, and Almarasan, in the spring, we observe a frost, temperature dropping down until -5 degrees. After that, in the May, warm and humid weather was observed, which created favorable conditions for disease development. Hot and dry weather in summer reduced disease activity, but favorable conditions return again in September. Little bit another situation in Alma Arasan. In Alma Arasan area, the situation was like I said, spring and summer were cool and very high humidity and precipitation. High humidity up to 93% and precipitation 159 millimeters. In Saragash, Tulkubas, and Merkem, warm and humid conditions in April. Also was frost in the spring, but extremely hot temperature in the summer. Hot and dry summer weather reduced disease spread, but increased plant stress and weakened the trees. And in the field, we observed different type of lesions and cracks on the bark on the walnut. At this slide, I want to tell about symptoms of bacterial diseases which we observed in the field during the field monitoring. The most widespread disease was walnut blight, caused by Pantoia agglomerans. It appears like has brown spot on the leaves, shoots, and fruits. Pseudomonas syringae was appeared like as wilting, and after that is lead for death of leaves and fruits. Pseudomonas aerosahibitans mainly affected the edges of leaves. Xanthomonas arboricola mainly affected fruits, but on the leaves we observed a lot of brown spots with a yellow halo. At this slide, I will tell about characteristic of fungal disease of walnut, which we observed on the field monitoring. Among fungal diseases, brown spot caused by Pantoia agglomerans was widespread. It appeared as brown spot on leaves, shoots, about Fusarium species. Fusarium proliferatum caused brown spot along leaf edges, leading to leaf curling and drying. Fusarium graminearum infected fruits. Fusarium salani caused black spots on young branches. Fusarium redolens, it is aggressive pathogen, causing tree weakening and wilting. And the last one, it is Fusarium incarnatum. Fusarium incarnatum caused kinkers on the trunk and branches. At this slide, I will describe about our field monitoring. We carried out our field monitoring from 2022 to 2024. We used a method of Sidov-Nagaltsova. We observed approximately 10 to 40 trees from one farm. We observed a tree from 4 sides, 4 branches. Almost we calculate 100 leaves from one tree and 25 leaves from one branches. After that, we're using formula described by Xi and Zhang. After that, using a scale 0.99 describing by G and Zhang for it calculate DI and DCI, disease incidence and disease severity index. As you can see on the picture, the main aggressive pathogen, it is Pantuia agglomerans from bacteria and Alternaria alternata from fungi. Alternaria alternata and Pantuia agglomerans acted like as independent pathogen in Almara, Sanaria, in Fazenda, Umihalica, Trikubas village and Merke village. Acted together like as mixed infection in Isik State Dendrological Park, Rural Farm Manshuk, Karadala Farm and Saraga Jersia. Speaking about mixing infection, also I want to say about Xanthomonas arboricola. Xanthomonas arboricola and Fusarium species, together with Pantuia agglomerans and Alternaria alternata, also acted like as mixed infection. Other Fusarium species was observed locally, and Agrobacterium tumefaciens, Pseudomonas aerosahibitans, and Pseudomonas syringae also was observed locally and like as independent pathogens. And disease incidence of Alternaria alternata was ranged from 30 to 50%, and disease severity index of Alternaria alternata was ranged from 10 to 26%. And Pantuia agglomerans disease incidence was ranged from 25 to 55, and disease severity index was ranged from 8 to 26%. At this slide, I will say about characteristic of isolated pure culture of bacterial pathogens of walnut. First of all, I will describe the method. We used sterilized plant fragments. After that, we homogenized in 1 milliliter phosphate buffer saline. After that, using Drygalski spatula, we apply on the nutrient agar. After that, after incubated, we transferred in the pure culture. And overall, we obtained 88 isolates. Pantuia agglomerans almost was obtained from stems and leaves and fruits. Agrobacterium tumefaciens from stems. Pseudomonas aerosahibitans mainly was obtained from leaves. Xanthomonas arboricola from fruits, and Pseudomonas syringae from leaves. At this slide, I want to say about isolates pure culture of fungal pathogens. First of all, I will describe the method. We used a wet chamber method. We also used a fragment of walnut, sterilized in 70% ethanol. After that, we used a wet chamber. We put these fragments in the petri dish with a paper, with a wet paper. After that, incubated and wait sporulation and mycelial growth. After that, we transferred this mycelium to the potato dextrose agar. Incubated, and after 7 days, we transferred in the pure culture in potato dextrose agar. We obtained 108 isolates. Alternaria alternata was obtained from stems and leaves, and the number of examined isolates was 40. It is 37% of total. Fusarium solani was obtained from stems. Fusarium proliferatum almost was obtained from leaves. Fusarium redolens and incarnatum was obtained from stems, and Graminearum from fruits. At this slide, I will say about identification pathogen using molecular genetic markers. For fungi, we use ITC reagent. For bacteria, we used 16S RNA. The PCR products were sequenced, and the obtained sequences were compared with reference sequences in GenBank using BLAST-N. Fungal isolates were also analyzed using the BOLT database. In addition, phylogenetic analysis was carried out in MEGA-12 to confirm the identification. We collected approximately 400 leaves and did a PCR analysis for molecular genetic markers. At this slide, I will say about pathogenicity test. For fungal disease, we used the mycelium and transfer this mycelium to wounded place of leaves and stems and fruits. For bacteria, we use a bacterial suspension. We apply this bacterial suspension also to the organs of walnut. After that, we measured the lesion size using ImageG program. All fungal and bacterial pathogens show pathogenicity except except Agrobacterium tumefaciens. Agrobacterium tumefaciens didn't show galls on the tumors. At this slide, I will say about genetic diversity and population structure. As you can see on the picture, principal component analysis PCA. Our wild forms situated separately and situated between Chinese and European clusters. And our wild forms has a unique structure. Wild Kazakh population also show unique genetic components confirming their genetic distinctiveness. Kazakh genotypes display mixed ancestry, indicating their intermediate position. At this slide, I will say about resistance forms. We take experiments for resistance against Pantoia agglomerans and Alternaria alternata, and identified the Miroslava, Avidio, Germisara, and Kazako, who shows high resistance and have score 0 to 1. This variety from Romania, Ukraine, and Moldova. It is introduced forms. At this slide, I will describe about evaluation fungicides against fungal pathogens of walnut. For bacteria, we used food poison technique method. In the petri dish with potato distress agar, we first of all pour fungicide suspension. In the middle, we transfer a mycelium of fungal. After that, we measured the growth of mycelium. And in the result, in laboratory experiments, dificonazole shows the highest antifungal activity inhibition grows by 92%. The second most effective fungicide was cresoxim-methyl with 73% inhibition. At this slide, I will tell about effectiveness of bacterial preparations against walnut pathogens. For bacteria, we used a food poison technique. We used a Miller-Hunton agar. On the Miller-Hunton agar, we pour a bacterial suspension, and on the bacterial suspension, we put a white disc impregnant with the bacterial preparations. And after that, we measured the inhibition zone with ImageG program. And in the result, the casumine with active ingredient gasutumin showed the highest antibacterial activity and can be considered like as most effective antibacterial preparations. And the Agrigent Plus with active ingredient gentamicin and oxytetracycline, and also demonstrated good efficacy, up to 80–90%. At this slide, I will tell about implementation recommendation for production. Based on the obtained result, we published practical recommendation for production and developed, including a set of agronomic, sanitary, and chemical measures. And in the farm of Mikhalicha, 2 hectares of disease-resistant walnut cultivars were established. At this, I will tell about economic efficiency using modern preparation against the main disease of walnut. As this table shows the effect of different treatment of yield, net incomes, and profitability. The highest yield was obtained with SCORE, difenoconazole, and followed by Casumin and Strobi. SCORE also provided the highest net income and profitability, 297%, making it the most economically effective treatment. And in conclusion, I want to say that it was a comprehensive work. We identified 10 pathogens. We observed and carried out field monitoring in 9 plantations. PCR confirmed the presence of key pathogens. Pathogenicity tests revealed a high degree of virulence in most isolates. We identify the resistance forms against Pantoia agglomerans and against Alternaria alternata. Also, we identify bacterial preparations and antifungal preparations. And in the end, we develop recommendations for protecting walnut plantations from fungal and bacterial diseases and implement our result in plantations. And we consulted all 9 farms. Thank you very much. Thank you for taking a valuable time and listening my scientific research.
Thank you very much. The presentation is concluded. Dear members of the Dissertation Council, questions may be asked orally or in writing. Are there any questions for the applicant? Please, do you have a question?
Well, dear members of the council, I have a few questions, if you allow me to ask. Dear candidate, my first question for you is, what novel insights does this study provide compared to existing international research on walnut diseases?
Thank you for your question. The novelty, if we compare with foreign, I think that it is our genetic diversity test with S&P markers. We collected 400 leaves. From 400 leaves, 173 leaves was examined in this method, and we obtained 143,000 markers with the Axiom G700 method. And because in the foreign we have in the Europe, Chinese clusters, but in the Kazakhstan was a gap. And I think that this help for the next genetics to fully have information for resistance forms.
Thank you for your answer. That was quite informative. My second question is, how feasible are the proposed plant protection recommendations under real agricultural conditions within Kazakhstan?
Thank you for your question. We carried out the field monitoring and laboratory experiments for identify fungicides and bacterial agents, and we identify these preparations that can be used in our conditions, in our natural conditions. And our advantage is that we identify the preparations that farmers can use specifically in your farms, in his farms. For example, in one farms we observed Pseudomonas syringae, and we gave Pseudomonas syringae on the pear. Pear was grown near with the walnut, and we gave advice to use antibacterial preparation together, not just on walnut, but on pear also. And for example, in the next farm, we know that they never used different type of fungicides or preparations, and we gave specific plans for treatment and agrotechnical sets. And our recommendation fungicides and antibacterial preparations registered in our country, registered in Kazakhstan, and I can say that our advice can be applied in Kazakhstan.
Do you have any questions? Dear dissertation consults, online, offline.
Yes, dear colleagues, hello. I have some questions. May I ask? Are you hearing me?
Please.
Yes. I have some questions. First of all, which pathogens do you consider the key ones for the southern zone of Kazakhstan? And on which current quantitative indicators is this conclusion based? Maybe step by step, okay? Please answer this question.
Key pathogens.
Yeah, which pathogens do you consider?
Yes. Yes, we carried out the field monitoring from 2022 to 2024 and identify 3 key pathogens. It is Pantuia agglomerans from bacteria. From fungi, it is Alternaria alternata, and also bacterial pathogen, it is Xanthomonas arboricola, because they give more disease incidence and disease severity index, and most aggressive, and we observed Pantuia agglomerans and Alternaria alternata almost from 7 farms from 9 farms.
And so, could you focus please quantitative indicators in this conclusion? How did you evaluate it?
How I evaluate? Yes, we used the disease severity index and disease incidence and disease severity index using a method described by Xi and Zhang. Also, we used a method described by Sedov and Nagaltsova. After that, we're using a scale of damage 0 to 9, and we also did a pathogenicity test. We also did examine on the resistance and re-isolation with coach postulate, and in the result, and also field monitoring observation, and in the result, we can say that Pantuia agglomerans and Alternaria alternata causes significant losses for farmers and the aggressive pathogen, a more widespread pathogen.
Okay, thank you for your answer. And the second question is, which walnut cultivars showed resistance under field conditions and what data support this?
We did experiments in the Manchuk rural farms and examined 23 introduced walnut cultivars for resistance and identified that Kazaku, the most resistant from pathogens and can grow in our conditions, in our natural conditions. After that, Miroslava, Ovidiu, Yarivsky, Chernovitsky, they have a score from 0 to 2% of damages. Score described by J and John.
Okay, that's enough. And the third question is, which preparations demonstrated the highest effectiveness in field trials, and what criteria were used to evaluate their efficiency?
From fungal pathogens, it was a score with active ingredient difeconazole and strobi with active ingredient chrysoxymethyl. Against bacteria, it is casumine, active ingredient cazumin, and Agrigen Plus with active ingredient tetracycline and gentamicin. We choose this preparation because this preparation show a high effectiveness in the foreign country, again walnut, because in our country we haven't registered pesticides. And in our laboratory and field treatment also these preparations show a high effectiveness. We chose this preparation also because, for example, casumine and Agrigen Plus has an antibiotic and we recommend use for first of all antibiotic. After that, for preventive measures, preparation with Cooper,
Okay, and I have another question. How do you explain the differences between laboratory and field effectiveness of the tested preparations?
Laboratory and field experiments is different. For laboratory, we have anti-sanitary and sterilized place and the controls. We can control everything. In the field, we have climatic conditions like as rain, frost, sunlight, precipitation, and the laboratory and field monitoring is different, but both of important for experiments.
Do you have any questions, colleagues? Online, if you finished, do you have questions online?
Sorry, dear colleagues, I have the last question. And what do you mean by local varieties? Do you refer to Kazakh breed or regionally adapted or zone cultivars? So it was about Kazakh and foreign varieties, or just about zone Kazakh or Kazakh varieties? Yeah, this question, please.
Yes, in our experiment, we used the local varieties, wild varieties, and 23 introduced varieties. Local varieties, in Kazakhstan, we have one adopted walnut forms. It is Tanka skarlupy, zinc shell zinc shell forms. It was registered in 1973 years, and it is a local forms. Wild forms was grown in Almarasan Grove, and 23 introduced that walnut forms was given from Moldova, Ukraine, and another country. Local forms are grown in Saragaj Jersiya, Karadala farm. In Manchuk rural farms, just introduced walnut forms, and in Silkale and in Isik Dendrological Park are grown Chinese cultivar Liao.
Please clarify. Did you mean local? It's Kazakh potato, Kazakh walnut varieties, right? Or different?
Yes, local forms. It is thin shell forms. Was registered in 1973 years.
Registered. So you mean
Пока
from
скорлупы. Thin shells.
so foreign and Kazakh varieties, cultivars, right? All together. Local. The term.
Yes, local.
Thank you for your answers. No questions.
Thank you.
Thank you very much, Vadim Tagirovich. Other colleagues, do you have questions for applicant? No?
I will have a question for Elmira. First of all, good morning, everyone, and greetings from Turkey. My question is, after this thesis, following this thesis, are any detailed molecular studies focusing on identification of strains related to these bacterial and fungal pathogens being considered for future research after finishing PhD? Is there any planning work for you?
And now we continue work with walnuts. Now we now we take experiments with Trichoderma harzanium. It is like as biocontrol, because before we used a chemical and antibiotic against the walnut. And we will continue our work on the walnut because we want to expand this area. We want to have a lot of walnut in our country, and we will continue to give, continue to do comprehensive work in this area.
Yeah, but my questions on determination of strains of these bacterial and fungal pathogens go deeply and determine what kind of strains, how many strains you have. For example, Panthea aglomerans or Xanthomonas arboricola patovar juglandis. This is very important for future screening of your local and international cultivars on Kazakhstan in different zones. So this is important point. Maybe later on you need to collect more isolates from different zones, walnut growing zones, and then try to determine using different molecular methods differentiate these strains. This is very meaningful. Maybe after finishing this, work.
Now we will continue to do identification of pathogens because every year pathogens is more and more, and give losses for farmers, and we now give consultation for each farmers from each rural farm, and I think that we start this way, and we continue, and we will work on this way, and we do not stop the identification pathogens using molecular methods, and we will use another method because a lot of pathogens now have a resistance, and another that we notice that a lot of pathogens was given before, now is we didn't find them. For example, Marsonia. And the interesting that Pantoia agglomerans can stop the growth of Marsonia. It is also interesting for us, and we will study this research.
Yeah, thank you. The other question will be on control of these pathogens, especially for bacterial diseases. You mentioned in your thesis, as far as I know, you used some antibiotics. It's very effective, but in practically, copper mixtures in other countries use very frequently. What about in Kazakhstan? Is antibiotics is very common using for bacterial disease control?
Yes, we give when we gave a recommendation, we just a second. When we give a recommendation, we give recommendation specifically for each farmers, and antibiotic can act it against Pantoia agglomerans and against Xanthomonas albaricola, which acted together in one farms, just in one farms. They can act it together. In other farms, they acted separately.
Yeah, why I'm asking this question, because after sometimes you get some resistance to pathogens that can be problem for controlling bacterial diseases. So in Europe and the United States, in our country, Turkey is forbidden using antibiotics. So maybe in next step, you need to get some, situation for using antibiotics instead of copper mixtures.
Yes, after time will be resistance for antibiotics, for example, for Casumine, for Agrigent Plus, and we try to find another fungicide and bacterial preparations. For example, now we are working, we want to work with Trichoderma hazarnium, like these preparations, and yes, we will search in other preparations also. But now for like as emergency, the Casumine and Agrigent Plus, I think is the best way, because in all farms, uncontrollably, we observed different type of pathogens, not one, several, bacterial and the fungal, Anthenaria species and Fusarium species, Pseudomonas, Xanthomonas.
Yeah, thank you.
Thank you for question.
Thank you very much, Hussein Basim, for your questions. Very good questions you give for applicant. Any question? Other our dissertation consuls? Allah bash. Yeah.
Okay, Almira, thank you so much. You started from field surveys to pathogenicity tests and then molecular analysis, finally with some recommendation. I appreciate good work. I just need to ask a couple of things. So because I'm not a conventional plant pathologist, I'm interested to know about these markers. You used ITS?
We use ITS for fungi. Yes.
Yeah, so let's talk. So ITS, if we talk about their reliability, and if we talk about their competency to differentiate two species of one genus, how efficient these markers are. For example, if you take example of Alternaria, Fusarium,
Yes.
then how efficiently these ITS markers can differentiate these two species?
After ITS markers, we use the bolt system, nucleate sequences. For example, for Alternaria, is a little bit hard to identify the species because Alternaria beach, Alternaria alternata, or Alternaria mali, and we give phylogenetic trees. We analyzed in a bolt system, in gen bank, for identification the patavars.
Okay, I mean, do you really need, if you're working with ITS, and if you feel challenged in identifying, differentiating two species of the same gene, do you need to test them some extra markers, some extra parameters?
Yes, we will use in other markers, not just ITS, because not one markers we have in the laboratory, but in our case, ITC region was applicable. But for example, now we give experiments with Apple also, and we use 5 markers for Alternaria species, for example.
Okay, I mean, you can further differentiate, not by using ITS, the second marker.
TEF1, for example, TEF1.
Yeah,
TEF1 is the best, yes. For Apple, we use a TEF1 also. For walnut, ITC is good.
Nice. Okay, and also in your thesis, somewhere you mentioned that your data of SNP. First of all, I want to ask, all this sequencing data, you did it yourself? You analyzed yourself? From where you got these services?
About SNP, this work was done with in our project, and not just me. It is a big team in yes, laboratory from in biology and biotechnology university with experts, Dilara Alexandrovna.
Okay.
Yes,
So
I was like a just assistant.
yeah, in your thesis you mentioned the SNP are helpful for the breeding of walnut. So what I see that these SNP does not really help, you could not integrate them, how they will be helpful for a breeder, for the walnut breeder. Can you just elaborate how they would be helpful? Because these are, yeah.
Except this PCA was analysis, just a second. Our wild walnut, walnut.
The snips from pathogens, right?
Yes,
Yeah.
genetic
How they
diversity.
would be helpful for a breeder?
For breedings, because in the world we have in the China, in the Europe, we also have genetic diversity results, but in Kazakhstan was a gap, and we find that our wild forms never was applied with the fungicides, bactericides. It has a unique structure, and I think that it will help for breeders in the future. And our wild forms situated in the middle between Chinese and European clusters. And I think it's very important because in the future we also have plans, for example, for introducing the new forms maybe, and it's very important for breedings.
And the last.
Alakbaş, sorry, could you talk to Mike because it's being recorded,
Yeah.
and your question should be recorded in the
The last.
Thank you.
And you did very interesting experiments, economic efficiency of modern preparations, and you found the profitability to 97%. I'm just curious to know how did you perform this experiment? I'm sorry, I missed it.
Yes. We calculate cost, net income, and profitability. We calculate, and the score shows the highest, yes, 297%, because it is after calculated disease incidence in the field. We carried out field experiments 2 years and calculate.
Was it replicated data?
3 replicators, yes, during growing season, yes. 3 times was applied fungicides, opening on the flowers in the middle, and after 30 days after applying, for example, fungicides. Yes, we calculated disease incidence, and using a formula of economic importance, we calculate.
All right, thank you.
Thank you for question.
Thank you very much. Any questions, colleagues? Please.
Thank you very much, Elmira. Very nice delivery your PhD thesis. So I had the same questions like Allah Baksh, and this is just for the consideration for future. I mean, on the profitability, it's a huge promise, you know, saying this kind of profitability. It would be great to see in the future just, you know, the other statistical parameters in this table, like, you know, the how deviate the data from the mean and the overall statistical significance. This is just a for consideration for future. Yeah.
Yes.
Do you have that one or?
Yes, we give a statistical analysis describing by Despechov. We also have an article with a statistical analysis, and using ANOVA, and after that we calculate LSD 0.05, and we use in our research one-way factor analysis and two-way factor analysis also.
Thank you very much. Yeah, it would be great to see in the presentation. Anyways, I'm sure it is in your PhD thesis. So I'm sure also this next question is, you know, about related to the literature review. I might miss that point in your presentation because I'm really sorry I didn't read the thesis fully from, you know, from the beginning to the end. So you are saying that this topic hasn't been studied in Kazakhstan at all. So you are the first one who is doing such
Yes.
kind of deep analysis on walnut.
Yes, it was a first comprehensive work regarding with identification and pathogenicity tests and fungicides. Regarding walnut, I made just one work, but it was about aflatoxins in the walnut, but not and the identifications diseases, and it is a different work. Our work is first in our country.
Sure. Okay, great. So if it is the first work, so I wanted to know, you know, I don't have really any idea about the, you know, the walnut in Kazakhstan because I work with wheat mostly. So how big is the walnut germplasm panel of Kazakhstan? So because you took some, you know, the introduced varieties and the wild species and the other cultivars from varieties or from other countries. So how big is the panel? Do we have our own varieties? So this is the first question. Second question is what about the breeding state of the breeding of walnut? Is someone doing breeding of these trees or?
In our country, Manchuk Jaksembina bring 23 introduced cultivars from almost Europe, almost in Moldova. In Moldova, the professor Kishinev cultivated this one, and she is first woman who started this work. And we have just 23 introduced cultivars in our country, local, and one Chinese cultivar.
Do we have our own varieties? So we don't have our own varieties, yes? Okay, sure.
We haven't else, but we implemented 2 hectares.
Okay, thank you. So the last question is, hypothesis, let's say, because when these days we talk a lot about the organic farming. So agricultural, the organic agriculture systems are in trend nowadays. So could you speculate what would be the first step if we start doing kind of organic farming on walnut? What would be the first step?
Yes, of course. How I mentioned before, we use now antibiotics and chemical preparations because we have, how I can say, is a bad situation in the all of farmers. All of farmers have 3 points of damages. And first of all, we use antibiotic and chemical, but now we did experiment with trichoderma and it is preparations named trichodermine, planris, and in the future also we will give recommendation use biological preparations. Also we also want to use a biological method, organic method. Yes.
Thank you. Any questions? Oh, okay, please.
From fungicides, it is score with active ingredient difeconazole. From bactericides, it is casumine with active ingredient cagasatsumine. Second one is antibiotic. First one impregnant in the shell of the walnut and how I can say fills impregnate in the shell and kills the members of the pathogens. And the antibiotic, as you know, can cause immediately. So we choose these preparations.
Okay, I have any question? Several questions.
Last. So
please.
what you are planning after your PhD? What you are planning after this?
Right now, we continue work with the walnut, with the apple, and I take participation in different type of competition, and I want to tell about our walnut situation for all, for international, in the international countries. Yes, and we will continue work on.
So you will continue work on walnut.
Yes.
So that's it.
Yes. Yes.
Okay, I have questions. How many area on Greece walnut in Kazakhstan planted?
In Kazakhstan nowadays we have 1,000 hectares of walnut. Almost the local farms.
Local farms and
It is almost amount 100 hectares.
100. Okay, next question. Do plant pathogens develop resistance to antibiotics? And what integrated pest management strategies would you recommend it for farmers? How many time treatments per season
Yes.
are optimal?
Yes, we gave recommendations for each farmer specifically and use the first of all antibiotics 3 times. Use like a preventive method, chemical pesticides with Cooper, like as preventive just and approximately 3 times. But we have plantations where growth of pathogens so big and individually was another amount.
Yeah, because antibiotic not good for.
Yes, because
resistance.
yes, it is harm and in the future maybe resistance. We know, but in the several countries we have so bad situation, and it is needed measures.
Okay, next my question about your decision implementation certificate. And here, no informative. only words you recommended. Which kind of you recommended here against diseases? Which kind of biological efficiencies in here don't information? You include here two implementation act certificate. For me, it's no information here.
Yes.
Farmers too.
Yes, we agree because it was a big work, and in implementation acts, we try to edit more information in the future.
Okay. It's enough, maybe, Alex? Okay, thank you, Mira, for your presentation. Okay, you can sit. The floor is given to the scientific consultant, candidate of agricultural science, associated professor of the Department of Fruit and Vegetable Growing, Plant Protection and Quarantine, Kazakh National Agrarian Research University, Gulsharia Kairova. Please.
Уважаемые члены диссертационного совета, так как я плохо владею английским языком, я попрошу вас разрешить мне выступить на русском языке.
Тогда нужно перевод, наверное, сразу, чтобы коллеги онлайн, которые
может,
офлайн сидят.
Ханат Ермекбаев перевести?
Да, кто? Вы же переведете, да? Давайте, ага.
Синхрон
перевод. Синхрон не получится, но топтап на кісі жалғасы.
Я поняла. Ну, одно из перспективных культур и ценным продуктом питания является для современного Казахстана орехоплодные культуры. И важнейшим вопросом выращивания орехоплодных культур является освобождение от вирусных, грибных и бактериальных болезней.
So I'm not a professional interpreter, but I will speculate. So the supervisor is saying that that this is in terms the work is perspective in terms of scientific and the practical point of view, and also about the food security. In that case, the identification of new diseases, strains, and is also as important as the first ones.
В результате проведенных исследований диссертантом впервые выявлены болезни, грибные бактериальные болезни, впервые изучена их распространенность, что подтверждает их распространенность в большей части обследованных площадей, что еще раз подчеркивает актуальность проведенных докторантом исследований.
During this PhD research, we have identified several bacterial and fungal pathogens for the first time in Kazakhstan, which shows the importance of the objectives of this PhD research.
Также докторантом проведена впервые оценка интродуцированных сортов и местных форм на устойчивость к основным грибным и бактериальным, ценный для, значит, как для селекционных программ, так и для производства.
Also, in this PhD research, the PhD candidate for the first time evaluated new introduced walnut varieties, which is very important to develop a local new walnut varieties.
На основе лабораторных и полевых исследований Эльмирой Исмагуловой Также впервые проведена оценка биологической хозяйственной эффективности некоторых фунгицидов и разработаны рекомендации для производства.
Also, Elmira has identified several effective fungicides, which in turn could be used in real practical breeding processes, also in real breeding practical side to grow these walnut trees.
Исследования проведены в рамках грантового проекта и имеют как научную, так и практическую ценность.
As this PhD research was conducted in the frame of grant funding, its objectives are very important for local walnut breeding.
По результатам проведенных исследований докторантам опубликованы статьи в Scopus в журналах с процентилем Q1, Q2, а также выпущены рекомендации для производства по болезням орехоплодных культур, то есть грецкого ореха.
As a result of this PhD research, Elmira has published several scientific publications indexed in Scopus and the Web of Science with high percentiles and the quartiles. Also, she developed a recommendation for farmers.
Эльмира Исмагулова в результате проведенных исследований она освоила современные методы фитопатологических, молекулярно-генетических исследований, также статистического анализа и обработки экспериментальных результатов.
During her PhD, Elmira has learned new approaches of walnut breeding, such as phytopathology and several approaches in statistics, how to analyze the data and interpret them. Also, importantly, she has learned new molecular genetics methods.
Хочу отметить, что Исмагулова Эльмира является состоявшимся молодым ученым, перспективным ученым, которая ее всегда отличают ответственное отношение к своей работе. Значит, она далеко смотрит вперед, очень старательная, скромная и перспективный молодой ученый.
So, as a PhD student, I'd like to mention that Elmira, she is although a PhD candidate, she is a established research scientist. She knows what to do and how to implement her results in future.
Также хочу отметить, даже вот после принятия работы к защите, она не успокаивалась, то есть подготовила несколько статей, в том числе и в Scopus, и подала на конкурс «Лучший исследователь 2026 года» и получила диплом, наградили ее дипломом первой степени. Это говорит о ее стремлении к науке, ее очень старательности и, в общем, стремление ко всему прогрессивному.
She's a progressive student, and I'd like to say that simultaneously doing her PhD, she also published several papers, as we mentioned before, and at the same time, she applied for local grant funding, and also she applied for the competition Best Young Scientist 2026, and she got a certificate of grade 2. if I could translate it right.
Считаю, что Исмагулова Эльмира, работа Исмагуловой Эльмиры соответствует всем требованиям Комитета по обеспечению качества в сфере науки и высшего образования. Диссертант заслуживает присвоения степени доктора философии по специальности защита и карантин растений. И я прошу уважаемых членов ученого совета поддержать ее как молодой ученый.
Also, I'd like to finalize saying that the PhD thesis and the work itself all are in accordance with the requirements of the Committee for Quality Assurance in Education and the Science of the Ministry of Science and Higher Education of the Republic of Kazakhstan. And I'd like to kindly ask members and the participants of the Dissertation Council supporting her candidacy. Thank you.
Спасибо за внимание.
Thank you very much.
Thank you very much, Rushan Spina. Next, the floor is given to the foreign scientific consultant, PhD professor, head of the Department of Plant Protection, Faculty of Agriculture, Akdeniz University, Turkey, Dr. Hussein Basim, please.
Okay, thank you. First of all, I would like to express my happiness at attending Elmira's doctoral thesis defense. And I will try to share my thoughts on doctoral thesis with you. The PhD dissertation by Ms. Elmira Исмагулова entitled Phytopathological Assessment of Introduced Walnut Varieties and Development of Protective Measures Against Major Diseases for the Southern Growing Zone of Kazakhstan represent a comprehensive and original scientific work devoted to an important and underexplored issue of modern horticulture. The study focuses on phytopathological monitoring, pathogen identification, and the development of protection strategies for Juglans regia under the agroecological conditions for southern Kazakhstan. The research is highly relevant considering the growing importance of walnut cultivation in Kazakhstan and the increasing incidence of fungal and bacterial diseases affecting productivity and orchard sustainability. The dissertation addresses the lack of systematic phytopathological studies and the absence of registered plant protection products for walnut in the Republic of Kazakhstan. The author's work contributes valuable data to national and regional strategies for sustainable walnut production and plant health management. The objectives of dissertation are clearly formulated and logically interconnected. The author employed a multidisciplinary approach combining field surveys, microbiological assays, molecular genetic identification using ITS and 16S RNA sequencing, and CMP genotyping and evaluation of fungicides and antibacterial preparations. The experimental design is methodologically sound, statistically evaluated, and demonstrates a strong comment of modern phytopathological and molecular techniques. The use of both field and laboratory experiments adds credibility and reproducibility to obtain results. The dissertation provides, for the first time in the Kazakhstan, an in-depth characterization of the phytopathogenic complex of walnut using microbiological and molecular genetic approaches. The study identifies key fungal and bacterial pathogens, including Xanthomonas arboricola patovar juglandis, Panthea agglomerans, Alternaria alternata, Fusarium solani, and others. The author developed diagnostic profiles for these pathogens and established the genetic diversity and population structure of Juglans regia cultivars and genotypes based on the CMP genotyping. This represents a significant scientific contribution to both applied and theoretical phytopathology. The results are presented clearly and analyzed thoroughly. the author successfully demonstrates differences in disease incidence and severity across cultivars and regions, identify resistant genotypes, and evaluated the biological efficiency of the modern fungicide and antibacterial agents. The findings have direct practical relevance for the orchard management and breeding programs. The discuss reflects a deep understanding of plant pathogen interactions and integrates the results into a broader international scientific context. The research findings have been implemented in walnut farms in the Almaty and Turkestan regions, confirming their practical utility. Developed recommendations contribute to improving walnut yield, reducing chemical use, and enhancing environmental sustainability. The outcomes of this study have significant importance for Kazakhstan's walnut industry and may be applicable to other Central Asian countries with similar climatic conditions. The research conducted by Elmira Ismagulova demonstrates her deep mastery of modern scientific methods, analytical thinking, and ability to carry out independent research work. The author has successfully performed a comprehensive set of experimental studies showing a high level of knowledge in the field of phytopathology and plant protection. The results of dissertation are reflected in scientific publications, including articles published in journals indexed in Scopus and Web of Science, which confirms the quality and international significance of the research conducted. The publication of the results in the reputable journals ensures recognition of this work by the international scientific community. Considering the relevance of topic, the scope and high scientific level of research work, as well as the practical importance of the obtained results for agricultural production, I believe that the presented dissertation fully meets the requirements of the Committee for Quality Assurance in the field of education and science of the Ministry of Science and Higher Education of Republic of Kazakhstan. In the view of the above, I recommended Elmira Ismagulova for the award of degree of Doctor of Philosophy in the specialty 8D-104 Plant Protection and Quarantine. Thank you. I like to mention that I have an undergraduate course now. I am already late. If you allow me, I need to leave from your meeting. And I like to congratulate Elmira and the other involving professors for this work also.
Okay, you agree? Thank you very much.
All right. See you then, everybody. Hello from and greetings from Turkey.
Okay, thank you very much. See you soon.
See you soon.
Okay, we continue. The floor is given to the first official reviewer, Doctor of Biological Science, Chief Researcher of the Laboratory of Mycology and Algology, Associated Professor of the Institute of Botany and Phytointroduction, Yelena Rahimova, please.
Allow me to present the review of official review for the dissertation submitted by Elvira Savetovna Ismagulova, entitled Phytopathological Assessment of Introduced Walnut Varieties and the Development of Protective Measures Against Major Diseases for the South Fruit Growing Zone of Kazakhstan, for the degree of PhD in the education program 8D 08104, plant protection and quarantine. Paragraph 1. The dissertation was carried out within the framework of the budget program 102, grant financing and scientific research under the sub-priority innovative biological research to improve the productivity and resilience of plant varieties and animal breeds in agriculture. The work was conducted under the grant investigation of the main walnut diseases and molecular genetic basis of the resistance of promising varieties to economically important pathogens. Paragraph 2. The work makes a significant contribution to plant protection and quarantine, and its importance is well articulated. Paragraph 3. The author demonstrated a high level of independence in planning and conducting field research, as well as in analyzing the obtained data. According paragraph 4, the relevance of the dissertation work is justified. The walnut, Juglans regia, is recognized as a highly valuable food commodity. And domestication of the walnut, commerce in antiquity, results in the development of numerous cultivars. These cultivars exhibit considerable variation in morphological characteristics such as fruit size as shape, pericarp hardness, internal septal development, and biochemical composition, among other parameters. The content of dissertation fully reflects its topic. The research goal and objectives correspond to the dissertation topic. The charter of the dissertation are fully logically connected with each other and are subordinated to a common goal. The results correspond to the objectives posed, and the conclusion followed from the results obtained in the analysis of a large experimental material. The author proposes new solution, principles, and methods that are well-founded and compared to existing approach. The comparative critical analysis is present both in the description of the identified fungal and bacterial species and in the assessment of resistance of walnut cultivars. Paragraph 5. The scientific results and provision are completely new, and novelty of dissertation work is confirmed by 3 highly rating publication and during the approbation of the work at 3 international scientific and practical conference. For the first time in Kazakhstan, a phytopathogenic component of walnut microbiota has been identified. The conclusions of the dissertation are completely new and follow from the results obtained from the analysis of a large amount of experimental material. Technical, technological, economic, and managerial decisions are particularly new and justified because walnuts have been cultivated for quite a long time. Paragraph 6. All main conclusions are based on compelling scientific evidence. Sufficient experimental data has been obtained to support the conclusions. Paragraph 7. There are 5 main provisions submitted for defense and characteristic for each provision are the position is proven, is not trivial, is new, the level of application is average since the applicability of the work is limited by the geography of region, proven in a number of top rating article. Paragraph 8. The methodology is described in sufficient details and substantive. All theoretical conclusions are supported by field and laboratory studies. The results of dissertation are obtained using both classical methods of mycology and phytopathology and modern methods of scientific research and methods of processing and interpreting data using computer technology. Theoretical conclusions, models, identified relationship and patterns are proven and confirmed by experimental research based on the Kazakh National Agrarian Research University and the Department of Plant Protection Faculty of Agriculture, Akdeniz University. Important statements are confirmed by references to relevant and reliable scientific literature. The references used are sufficient for literature review. The list of references includes 156 titles. Paragraph 9. The dissertation has theoretical significance, which is comprised of conducting research on the phytopathogenic complex of walnut, determining the species composition of fungal and bacterial pathogens, creating diagnosis profiles of pathogens, and the developing scientifically recommendation for protecting walnut plantation from diseases. The dissertation has practical significance for Kazakhstan agricultural sector, phytosanitary monitoring system, database of principal pathogens, recommendation of stable varieties. Yet their practical utilization is restricted often to geographical specificity. The proposed practical recommendation are partially new for the peasant farm, Manshuk and Fazenda. Paragraph 10. The writing quality is generally high, but there are occasional stylistic errors and grammatical types. The academic style is consistent. Paragraph 11. There are 3 comments on the dissertation. 1. When the species of fungi or bacteria are first mentioned, the author who described the species are not indicated. 2. The micrographs do not indicate magnification. 3. There are some stylistic errors and grammatical types. The comments is no way diminish the value of this dissertation. The doctoral students' articles demonstrating a high scientific standard are relevant to the research topic and reflect the key aspects of the dissertation. In article published in peer-reviewed journals, The author provides the detailed description of the results, data analysis, and discussion of the conclusion, confirming the scientific value and novelty of the work and demonstrating the deep understanding of the problem and the ability to apply modern analytical methods. A total of 9 scientific works have been published based on the research. Among them, 2 articles were published in journal Q1 and Q2. My decision, the dissertation work of Elmira Savyetovna Исмагулова, entitled Phytopathological Assessment of Introduced Walnut Varieties and the Development of Protective Measures Against Major Diseases for the Southern Fruit Growing Zone of Kazakhstan, meets the established requirement of the rules for the award of degrees, and its author, Elmira Savyetovna Исмагулова, deserved to be awarded the degree of Doctor of Philosophy in the education program 8D 08104, Plant Protection and Quarantine. That is all. Thank you for attention.
Thank you very much, Elena Vladimirovna. The follow is given to the applicant to respond to reviewer comments, please.
Dear reviewer Elena Vladimirovna, thank you very much for your valuable comments. All the mentioned issues have been carefully addressed. The authors of fungal and bacterial species are now indicated in their first mention. Magnification has been added to all micrographs, and the manuscript has been thoroughly serviced to correct stylistic and grammatical errors. Thank you.
Thank you very much. The floor is given to the second official reviewer, PhD Associate Professor of the Center of Excellence in Molecular Biology, University of the Punjab, Lahore, Pakistan. Allah Bash. Please.
Come to me. So this thesis by Almira, it did both work. It provided knowledge in the form of international publications in local and international journals. And on the other side, it provided recommendation to the farming community. So in either ways, this thesis is well structured and well written with all the information and introductions and literature review. And also it aligns well with all the priority areas of the government. So like mentioned in the literature review, the thesis addresses very well the gap in walnut studies, especially in context of plant pathology. So the thesis had field surveys, identification of pathogens, the proven of postulates, and then the utilization of the molecular markers that was on spot really did well to identify the strains. And then, the resistance studies, and finally the recommendations, all of these, the work, it shows the significance of the thesis in its true spirit. If we talk about the applicant, it was a really well-structured and relevant study. It addressed an important problem. in walnut diseases and their management. Of course, the thesis demonstrates a good combination of field investigations, laboratory experimentations, modern molecular techniques, which strengthen the overall scientific rigor of this thesis. The topic is timely and well aligned with the current agricultural priorities of the Republic of Pakistan, and the results have both scientific and the practical significance. The candidate has shown the ability to conduct independent research and produce publishable results in internationally renowned journals, which is worth appreciating. And when we talk about the scientific level of doctoral candidate articles, the articles are well demonstrated in SCI expanded and Scopus indexed. These publications provided a really good knowledge for the scientific community. It included articles, of course, in SCI and Scopus indexed. And also another thing which we disseminate is to present our research work in our conference proceeding, and that work was really done by the candidate, and the work was presented in two conference proceedings, and overall it shows that research findings are of acceptable quality, and quantity, visibility, and relevance to the broader scientific community. So after going through the dissertation work and outcome of the thesis in the form of research publications, a dissemination of work in conference proceedings, and all the practical recommendation for the farming community by the candidate, I believe the candidate deserves the award of degree. Thank you.
Thank you very much, Alakbach. The floor is given to the applicant to respond to the reviewer comments.
Dear reviewer Alakbash, thank you for your valuable comments. All remarks have been addressed in the revised dissertation. We strengthened the comparison with international studies and clarified the scientific novelty, especially for the conditions of southern Kazakhstan. The conclusions made me more careful and also added in limitation statements, nothing that further multi-year and multi-location studies are needed. The literature reviews was updated with the recent international sources, and the practical recommendations were revised according to the local phytopathological and agroclimatic conditions of southern Kazakhstan. Thank you very much for you.
Thank you. We will now proceed to open discussion. Who wishes to express their opinion? Please. Dissertation Council on online, offline, please.
I'd like to say a few things, if you allow me.
Please.
Yep. Honorable Chairman, members of the Council, colleagues, and of course the candidate, I value your time and try to make it brief. Overall, it's a real pleasure to evaluate this work. Expanding walnut production in Kazakhstan has a huge economical potential, but as we know, the pathogens are a major bottleneck. This dissertation tackles that problem head-on with both scientific rigor and practical solutions. I want to quickly highlight three things that really stood out to me. First, the methodology. The candidate did an excellent job bringing classic field pathology with modern molecular tools. Using ITS and 16S sequencing gave us a definitive DNA back profile of the pathogen we are actually dealing with in the cells. In this matter, I also agree with today's comments from our Turkish colleague Hussein Basim and another colleague Allah Bach on expanding the molecular markers, using some more other genes for the molecular identification. And the candidate confirmed that she started using TEF1 and other genes, which is a good news. The second is genetic work using SNP genotyping to evaluate the walnut cultivars, elevates these from standard crop protection to something that will genuinely guide our future breeding programs in Kazakhstan. And finally, the field trials, taking this out of the lab and turning it into field level economic metrics means our local agronomists and farmers walk away from with scientifically backed protocols that they can use right now. So overall, this is a mature, well-rounded piece of science. The candidate has shown a deep grasp of disease dynamics, genetics, and agronomy. I highly commend this contribution to our field, and I fully support a successful PhD thesis of the candidate today, Elmira Ismagulova. Thank you.
Thank you, Dr. Kazbek, your opinion. Next. Wishes.
Hello, dear colleagues.
Okay, please.
Yeah, I'd like to say a few words. It was very interesting. First of all, I want to cheer the chair board and dear colleagues. It was very interesting to listen such an interesting dissertation thesis because I'm not a specialist on walnuts, but I'm glad that some research in Kazakhstan is ongoing, and I hope that in future there will be some deeper research on breeding some local varieties of Kazakhstan agroecological conditions. And I wanted to say that, yeah, I had some questions, but my colleagues asked them, so I got some answers for my questions, and I'm very appreciate. Thank you.
Thank you very much, Elena, Dr. Elena. Other colleagues, it's enough, maybe? It's okay? Thank you. The closing remarks are given to the applicant, Elmira Sovetovna Ismagulova. Please, conclusion.
First of all, I want to express my special appreciation to my university, Kazakh National Agrarian Research University, as well as to all professors and staffs who contribute to my education and professional development. I would like to express my deep gratitude to my scientific supervisor, Kairova Gusharan Sapayevna. She guided me throughout this entire journey, supported me during challenging moments, and provided invaluable direction in my research. I consider her the best supervisor one could ever wish for. I also would like to thank my international supervisor, Hussein Basim, for his support and assistance at all stages of my work. He is truly one of the best international supervisors whose professionalism, kindness, and willingness to help made a great contribution to my research. I would like to acknowledge the mentors and specialists with whom we conducted our research, the staff of the Kazakh Research Institute of Fruit and Vegetable Growing with Savle Khorabaeva in the Institute of Plant Biology and Biotechnology, Zarpa Bisenovna and Dilara Alexandrovna, Gulnaz Kamirdanovna. I am especially grateful to Professor Sergei Nikolaevich, who supervised the field experiments and provided invaluable assistance and implementation. I also deep gratitude all the staff of the Kazakh Research Institute of Plant Protection and Quarantine, named after Jaskin Jimbaev, where I compared my internship, for their support and professional guidance. I would like to extend my gratitude to all of the heads of farms where I conducted monitoring on their cooperation and support. In particular, I would like to thank Manshuk Jiksenbekova, as the main part of the trail was carried out on her farm. I would like to express my deep gratitude to everyone present today at my dissertation defense today, and to those who took their valuable time to listen to my work. Thank you for your attention, support, and time.
Thank you. You can say it. Okay, thank you. That all, dear members of the dissertation council, for purpose of conducting and the secret ballot in accordance with the established regulations. The following composition of the counting commission is proposed. We will be choice Ермекбаев Kanat and Елена Рахимова Allah Bash. Let us vote, colleagues. Members of the counting commission, please proceed with your duties. Members of the council, please cast your votes. Остальных прошу покинуть зал. Так, председатель Ермекбаев Канат. Онлайн, которые сидят, тоже давайте проголосуем, члены диссертационного совета.
А вы нам ссылку отправьте, пожалуйста.
Сейчас. Уважаемые члены диссертационного совета, просим при голосовании ставить только баллы, да? Один ответ. Ага, можете ознакомиться с положением.
So we are kindly asking, when you vote, you need to give a score underneath. So the link hasn't been provided yet. So, Allah Baksh, you can look at the screen. So you have to choose those marks. Then you have to choose one of those four. Then add a mark, which the ball қою керек, yeah? So you need to give a score basically. So from 0 to 100, yeah, 0-ден 100-ге дейін. Ne?
Okay. The floor is given to the Deputy Chair of the Counting Commission, Dr. Emrekbaev Kanat, to announce the results of the secret ballot.
Hello, everyone, again. Thanks, the members and the participants of the Dissertation Council and the chairman of the Dissertation Council. Let me read out the protocol 1 of the meeting of the Counting Commission elected by the Dissertation Council, dated 29 April 2026. The meeting of the Counting Commission of the Dissertation Council for the defense of dissertations for the degree of philosophy, degree of Doctor of Philosophy, PhD, in the field of training 8D081 crop production, soil science and agrochemistry, plant protection and quarantine. Specialists 6D080800, soil science and agrochemistry, 6D081100, plant protection and quarantine. Educational programs 8D08102, soil sciences and agrochemistry. 8D08104, plant protection and quarantine, was held at the Kazakh National Agrarian Research University. To count the results of the secret ballot in electronic format for making one of the following decisions on the defense of the dissertation by Elmira Ismagulova on the topic phytopathological assessment of introduced walnut varieties and the development of protective measures against major diseases for the southern fruits growing zone of Kazakhstan. For the degree of Doctor of Philosophy, PhD, the Counting Commission was elected in the following composition. Chairperson, Khanat Abayevich Yermekbaev, it's me. Members of the Commission, Elena Rahimova and Allah Baksh. The composition of the Permanent Dissertation Council was approved in the number of 4 persons. Temporary members of the Dissertation Council were included depending on the topic of the dissertation research, with additional voting rights in the member of 3 persons. 2 official reviews took part in the secret ballot. 8 persons participated in the secret ballot. So, number of elected ballots sent was 8. Number of unsent ballots 0. Results of the secret ballot. First, to award to the degree of Doctor of Philosophy, PhD, 8. To send the dissertation for revision, 0. To send the dissertation for re-defense, 0. To refuse awarding the degree of PhD, 0. So invalid ballots, 0. Evaluation of the defense in percentage, 93. In grade letter, A-, 0.6 points in 3.67. Traditional grade is excellent. Chairperson of the Counting Commission is me. Then members, Elena Rahimova and Allah Baksh. Thank you very much.
Thank you very much. We will now proceed to the discussion of the draft and the adoption of the Council's conclusion on the dissertation of Elmira Sovetovna Ismagulova. Does anyone have proposals? Do you have and by hand conclusion project?
Yes, we do have it.
Okay, please. Dear colleagues, please let me now introduce evaluation of the defense. 93%, letter grade A minus, points 3.67, traditional grade good, excellent. Dear dissertation council, we need to classification criteria. First, nature of dissertation results. Which point we choose? Please recommend it for us.
(1.1)
Solution of problem of significant importance for the relevant field of knowledge. 1.1 or 1.2 presentation of scientifically grounded technical, economic, or technological developments ensuring the solution of important applied problems. Which point?
I would say 1.1.
Yep, the first one.
Yeah, we can go for 1.1.
1.1.
Allah Baksh. Yeah, 1.1.
Okay. Next. Level of novelty of dissertation results. 2.1. Results are new. 2.1. Okay. Third, value of dissertation results. 3.1 high, 3.2 satisfactory, 3.3 unsatisfactory.
3.1.
I would say high.
3.1 high. Okay.
Yeah, high.
4. Connection of the dissertation topic with planet research. 4.1. The topic is included in state and regional scientific and scientific technical programs or international research programs. 4.2. The topic is included in a fundamental research program, sectoral program, or plans of scientific organization and a higher educational institution. 4.3. Innovative work. Программа кірді ме? Грантовый проект.
Which one would you choose? 4.1? Yeah.
Это государственные, научные и научно-технические программы.
It was internal for the project. It means 4.1, yeah? Not program.
Государственные, да. Тогда 4.1. 4.1, okay? Next. Level of implementation of dissertation results of applied significance. 5.1 at the international level. 5.2 at the intersectoral level. 5.3 at the industry scale. 5.4 within the organization.
Yeah, 5.3 or 4.
Масштабах отраслей. 5.3. Yeah. Next, recommendations for expanded use of dissertation results of applied significance. 6.1 requires expanded use. 6.2 does not require expanded use. 6.1.
6.1.
We
Okay.
believe it does require.
Thank you. The dissertation council has resolved to accept the conclusion of dissertation council in accordance with the regulations on the dissertation council. Second, based on the public defense of the dissertation by Исмагулова Elmira Savetovna on the topic phytopathological assessment of introduced walnut varieties and the development of protective measures against major disease for the southern fruit growing zone of Kazakhstan, with a mean defense score of 93%, to consider that the dissertation meets the requirements for a Doctor of Philosophy, PhD, dissertation on the educational program AD 08104, Plant Protection and Quarantine. 3. In accordance with the model regulations on the dissertation council of the Committee for Quality Assurance and Control in Education and Science of the Ministry of Education and Science of Republic of Kazakhstan, and the regulation on the dissertation council of the Kazakh National Agrarian University, Research University, to award Исмагулова Elmira Savetovna the degree of PhD and the educational program AD08104, Plant Protection and Quarantine. Thank you very much. My congratulations.
Thank you very much, everyone.
Yeah. Thank you very much, everyone.
Thank you very much, colleagues.
Bye-bye.
Bye.
Thank you.
Thank you. Good luck, colleagues.